My name is Chauhan. I am a medical liaison with NS Pharma, and today I'm excited to share a presentation I shared in person at the PPMD conference. This is going to be an overview of Viltepso, which is an approved product for exon 53 skipping. And we'll just be going over some data, and we're excited to share 4 years of data, actually. Just a disclaimer that I may make some forward-looking statements in this presentation. If you're unfamiliar with our company, we're actually based in New Jersey, in Paramus, New Jersey. We're under umbrella of a bigger company, Nippon Shinyaku, and they are located actually in Japan. They have over 100 years of experience, and it's not just limited to DMD. When we look at Viltepso, who does it target? I think this pie graph does a good job of showing the different exons and the percentage prevalent in the population for the mutation. So today we're going to be targeting exon 53. As you can see, it's roughly around 8% of the population of DMD boys. And just to say, this is a once weekly infusion over 1 hour. So for today, I'm going to be showing you some data. I want to keep your attention on the left side, these two trials. So first, I'll be covering a little context about our phase 2 trial and then showing you some data that we were able to gather over 4 years. So first, I'll start with our original study, which is 20 to 24 weeks. What did we look at? Primary outcome was looking at dystrophin induction. Well, of course, with any clinical trial, you're going to see some safety, pharmacokinetics, and some tolerability statistics as well. As far as secondary, we just looked at different ways you can analyze dystrophin. And we looked at some clinical tests that many of you may already be familiar with. It's just time to stand, time to run, walk, time to climb, and those tests. So after 20 to 24 weeks of treatment, we did a muscle biopsy. And in the boys, the 8 boys specifically, we saw at a baseline, they started with 0.6% of normal dystrophin. After that treatment period of 20 to 24 weeks, we saw an average induction of 5.9%. And we were very happy with this result, since at this time, this was the highest percent of dystrophin induction in a exon skipping clinical trial. And I want to say all patients showed an increase in dystrophin levels. So that was a quick overview of our original phase 2 20 to 24 week study. Now I'm going to share some data looking at 4 years of those same boys. But before I move on, also want to note that after this trial completed, currently we're still ongoing our phase 3, which is the Razor 53 trial. And that's a global study, which is going to be our confirmatory trial. And that one is placebo-controlled. In addition to that, we also have another trial going on, which is our Galactic 53. And this trial is interesting because we're actually looking at boys in an older population. So they have to be at least 8 years of age. And we'll be looking at some clinical trial measures such as pulmonary data as well, in addition to muscle tests. So looking at the 4 years of data, first I'll start with the time to stand. And this is represented here in seconds. At the bottom on the x-axis, you'll see it goes up to 205 weeks. On the y-axis here, this is looking at the change from baseline in seconds. The blue line are the boys that were on Viltepso, and the yellow line was the natural history group that we compared to. So just looking at this, you can see over the first 2 years, we see a maintenance of function. And then the following 2 years, what we see is a significant delay in disease progression. And what I mean by that is, if you just look at the raw values up to 4 years, you can see boys that were on the natural history group. After 4 years, their time to stand increased by 8.5 seconds. When we look at boys that are on Viltepso, we see the raw value that the time to stand increased by only 2.5 seconds. And we know in this community, a maintenance is a win for families. Additionally, if we look at time to run walk 10 meters, a little bit of the same story is shown as well. Actually, here in the first 2 years, you may see an improvement in function. And then we see that the longer the boys are on therapy, we see a delay in disease progression. And then time to climb. This one was a little bit more parallel with the natural history group, but nonetheless, you're still able to see a difference over the 4-year time course. Of course, we know now exon skipping agents have been shown to have a very favorable safety profile. When we looked at our study, we saw that there were no participants that experienced any serious treatment-related adverse events, no patients discontinued, and there were no deaths in this study. So in the original group, we asked all the boys, do they want to continue for 4 years? And all of them happily accepted to continue into the long term. And finally, folks, today we saw some data on exon 53 trial, which is our Viltepso trial. That has been granted accelerated approval here in the US and Japan, but we're not stopping there as a company. As you can see, we have multiple therapies in the pipeline. Next, we have exon 44, which we were able to present a little bit of data and some information at the PPMD conference. And then next, we plan to initiate exon 50 trial as well, in addition to 45, 51, and 55. And our goal here is to cover as many patients as we can, and we're happy to take the learnings from this Viltepso trial and move it along with our other therapies. Finally, this is just a thank you to all the patients and the community. We understand there's many options here in the field, but we really do want to thank the community for your participation, because none of this would be possible without you guys. And if you have any questions, it's regarding access. We do have an in-house team, and that's called NS Support. The contact information is here for you, but once again, we're here for families, and there's a lot of things that families have to worry about. And the last thing we want to do is make it a barrier for access to treatment. And with that, thank you all for your attention.